Hi there and welcome to another biology lesson. Today we're going to be looking at protein synthesis. We're going to look at this a little bit different. What I'm going to do is I'm going to give you the brief overview first. I'm going to go through the diagram, then I'm going to take you through it in a little bit more specifics, and then I'm going to bring you back to this diagram, so we'll recap it at the very end. So here we have a very, very basic animal cell. I haven't stuck in any extra organelles or anything for you, just the things that we need. So protein synthesis, we're going to go through the primary basics of it in a second, but basically I want to know how our cells produce proteins. We'll go through what exactly they are and where they're used and their jobs and everything later on. So how does a cell produce protein? It all starts with DNA. Remember, DNA is our code for absolutely everything. Parts of that DNA are transcribed, meaning they're copied, and they're turned into messenger RNA, so mRNA. These mRNA strands look like little pieces of DNA, but only one side of the DNA strand. So mRNA, a little piece of DNA. Have a quick jump over to our diagram over here. We've got our DNA here, that's that blue squiggle in the middle, representing our DNA. We have the mRNAs represented by the pink. So you can see that when we get from DNA to mRNA, the DNA actually unwinds and separates, and then mRNA forms using the template of the DNA strand. Once that mRNA is made, it is modified slightly with bits of that mRNA strand removed and then some tails added to the end of it. After that point in time, the mRNA, the modified mRNA, then moves to the rough endoplasmic reticulum, where it meets with a ribosome. Now, ribosomes are pretty much they're our protein manufacturers or our protein builders. The mRNA is, the ribosomes then read the mRNA 3 nucleotides at a time, because 3 nucleotides make an amino. And so 3 nucleotides at a time. Once that has made read the 3 nucleotides, it knows what amino acid it needs, and there is another form of mRNA, but it's RNA, sorry, it's in the form of tRNA, which is a transport RNA. And what it is it's shaped a little bit differently, but I'll explain that in a second. And these guys here carry the amino acids. They're responsible for transporting the amino acids to the ribosomes so that they can be put together to make a protein or a polypeptide chain that then turns into a protein. So as the ribosomes read the mRNA strand that's going through it, that's been manufactured in the nucleus, as it reads one block of mRNA, it will then signal out, and the tRNA will then bring the amino acid to the ribosome, where it will start to put together the amino acids into a chain, which we call a polypeptide chain. When the ribosome has finished reading the entire mRNA strand, so once it's worked its way from one end to the other one, the mRNA strand drops off, and so does the polypeptide chain. And then the polypeptide chain basically folds and reforms itself and joins with a couple of other chemicals in order to become specific types of protein. So that's our rough guide. So the key things for us to note here is that we start with DNA. We then create an mRNA using the template strand of DNA. It's modified a little bit inside the nucleus. When it moves to the endoplasmic reticulum, where it meets up with the ribosome reads the mRNA strand. The tRNA molecule is used to bring the amino acid to the ribosome, and the ribosome sticks the amino acids together to create a protein. So something else you can gather from this is that proteins are formed from or built from amino acids. So many amino acids together make a protein. All right, let's have a look. Okay, so we have a fair bit going on this page, but we are going to work through this nice and succinctly. We've got a bit of a catch up here, and we're going to build up a little bit of base knowledge before we start over with our proteins. synthesis, and we're going to work a little bit this down the page before we head up and around and finish off over here in the corner. So focus one piece at a time. A couple things we need to recall. A gene is a segment of DNA that controls a specific characteristic. So genes are coded DNA, all right? Not all parts of our DNA are coded. Genes are the ones that have actually a job, or they're specific to a characteristic. The word genome refers to all the genetic material found in the body in a standard body cell. So the genome is found in all standard body cells except for red blood cells, and I'm going to underline that because that is a little bit of a difference, a key difference, I should say. All except red blood cells and gametes. So gametes are an obvious one because they can only have half of the DNA that a normal cell has. But red blood cells don't carry any, and they don't carry any because they actually don't reproduce, and they don't have cell nucleuses. So two cells that don't have a complete copy of the genome. That means, so to put that into perspective, or to help you understand that even further, that means that all of these cells that have a complete copy of the genome are able to become, or they have the instructions to be any cell that the body requires. So every single cell in eye cell will have inside of it also the instructions on how to be a brain cell, on how to be a skin cell. So like all of the instructions for every cell exists inside each standard body cell except for the red blood cells. A cell, therefore, why does an eye cell look, or a skin cell look like a skin cell? Why doesn't it exhibit all of the characteristics of the entire genome? And that's because these cells, each cell only expresses the appropriate genes for what it's supposed to become. So the cell, when it's in the process of being formed, will be signaled by a variety of different stimuli. And those stimuli will then sort of educate it or train it or make it known as to what it needs to become. And so certain genes, or if it's got, these ones are going to be getting the cells through mitosis. So those are obvious, they're going to become identical daughter cells. But in the formation, or in the early formation, when we're talking about stem cells and things like that, these stimuli might be related to location of that actual cell. So as to what it's actually going to become. So that's our quick recap at the top here, and we've built on it just a little bit through here. We're ready to talk about protein synthesis. So protein synthesis. Proteins. Particular proteins are constructed by particular arrangements of particular amino acids. So this is what we spoke about briefly before. Proteins are built out of amino acids. Amino acids, there are 20 of them. So 20 amino acids are used to construct all the proteins in your body. Each amino acid is coded for by a sequence of 3 nitrogen bases. Now we know your DNA is constructed of nitrogen bases, and we know that they are adenine, thymine, guanine, and cytosine. So 3 nitrogen bases in a row code for a particular amino acid. A particular amino acid, 1 out of 20, in fact. So what that means is, we'll go this way first. There are 64 possible sequences. So with 4 letters, there are 64 possible sequences that can be created. Out of that 64, 61 of them will code for aminos. That means there's some double up. We've only got 20 amino acids, and we have 64 combinations, so 61 of them will code for the 20 amino acids, or one of the 20 amino acids. So that means then 3 codes out of those 64 are used for starting and stopping signals. So I have recently actually just read a really nice sort of example for it. They're kind of like the punctuation in the writing of or creation of a protein. So 61 sequences to create 20 amino acids, some double ups there on that. So an example of this might be, if I have an adenine, 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 that code will actually code for the same amino acid as adenine, adenine, and guanine. So both of these code for phenylalanine, and that's just one of our examples that we have. There will be many, many more double ups for that one. All right, so protein synthesis. When coded DNA is processed to build a protein, that is what protein synthesis actually is. When coded DNA we also know is a gene. So when a gene is processed to build a protein. Examples of proteins are hormones, enzymes, antibodies, structural components of cells. So these are just some of the examples of proteins and where we find them and what they do. So let's have a look at the process of protein synthesis. Step number 1 in the process of protein synthesis is transcription. In this part of the process, the code is copied into another nucleic acid called messenger RNA or mRNA. So we spoke about this in our diagram. I'll quickly bring this one back over. We're talking about inside of the nucleus of the cell. This bit here we're actually going to call transcription. So I'll see if I've got my red pen here. Transcription occurs inside of the nucleus. So transcription on the inside of the nucleus. Put this one back. So it's key to note that DNA never ever leaves the nucleus. The mRNA does, but the DNA doesn't. So only copies do. In the process of transcription, the DNA actually unzips and separates, similar to when we have DNA replication, but it actually will unzip and separate. Then RNA polymerase will read the gene and attach the complementary bases or nucleotides to the DNA. So this is the DNA unzipping here, and we have the RNA polymerase that's reading the one single strand of the DNA and collecting the relevant complementary bases. So you can see that C and G, G and C have all been connected in T and A, but there's a little bit of a difference here around the A and what it connects to. So that's the next thing. In RNA or RNA polymerase, when RNA polymerase is actually attaching the new nucleotides, there is no thymine. So instead of attaching a thymine to adenine, it will actually attach a uracil to adenine. So that little side note is here. So RNA, no thymine, instead pairs uracil with adenine. Okay, so once the RNA has completed is complete, it will detach from the DNA, and the DNA will then rejoin back together. So once the RNA has been, or mRNA has been fully made, it will detach from that template strand, and then the two DNA strands will rejoin. The DNA amino codes are the next thing. So what has been coded here is now separated and on its own. So what has been coded? We know that it's a section of DNA. We know it's a section of DNA that's coded, and we know that it's a gene. But what has been coded is going to be used to determine what aminos need to be made, or amino acids, in order to make proteins. So in DNA, the amino codes are called triplet codes, and they're called triplet codes because it takes 3 bases to identify an amino acid, or to signal for an amino acid to be brought for protein building. So I've copied this strand out here, and I'm looking at the blue is being the DNA strand. So the blue DNA strand that has been copied into mRNA is ACCGTA. The blue strand, and those blue letters that are being used to create that are used to create the mRNA strand, are called DNA amino codes, the DNA amino codes are called triplet codes. The complementary mRNA strand that is created, also within those groups of 3, are called triplet codons. So these triplet codons are just the complementary pairs of the triplet codes, and each group of 3 will reference a particular amino acid. So I'll try that one more time. Each group of 3 nucleotides references a particular amino acid, which we spoke about over here. Okay, so it's at this point where it depends on the type of organism as to what actually happens next. If the organism is a bacteria, the mRNA is ready to become a protein. So that mRNA will then be moved out of, if this is a bacteria, the mRNA will then be moved out of the cytoplasm, where it will be read by a ribosome, and a protein will be put together by the tRNA, bringing over the correct or the relevant or applicable amino acids. But if the organism is a eukaryote, the mRNA is going to be split. So I'm going to reference this picture just here soon. So the mRNA is going to be split into exons, which are coded parts of that mRNA strand for particular proteins, aminos. And the other side is going to be, the other bits that are going to be split out are introns, and these are the non-coded bits. So in this process, what we're doing here, or what's actually happening is that the exons are going to remain and the introns are going to be removed. And the exons are going to be joined together. So we can see in this diagram, we've got our exon here and an exon here and an intron in the middle. The intron is removed and the exons are rejoined. When they've rejoined, they're now considered to be mature mRNA. So mature mRNA, but it's still not ready to leave the nucleus and go to the ribosome yet. So before it can do that, extra nucleotides are added at the beginning and at the end. Sorry, not at the beginning, at the end. The extra nucleotides that are on the ends of these mRNA strands are removed, and caps and tails are added to them. So they end up like this. So caps and tails are added to them, and the reason for that is that it makes it easier for them to bind to the ribosome so that it can be read. Makes them a bit more stable. After the caps and tails have been added to the mRNA, they are able to, the mRNA strand is now able to leave the nucleus, where it will connect with the ribosome. And a quick recap, the ribosomes are the protein builders. So these are the readers of the instructions, and basically the foreman that organizes, or the manager that organizes the aminos to be transported to the factory to build the proteins. So they assist, after this occurs, the ribosomes read the mRNA one codon at a time. So quick recall on what a codon is. A codon is the group of 3 nitrogen bases that codes for a specific amino acid. So 3 nitrogen bases that code for a specific amino acid. So the ribosome is there as its, if your mRNA strand is there and it's being read by the ribosome is going to read it 3 nitrogen bases at a time. Once it gets to those 3 nitrogen bases, it's going to wait until it has received the correct amino acid. At this point in time, once the mRNA has read the first codon, the tRNA, so the transport RNA, will bring the amino acid to the ribosome. Now, a little bit on transport RNAs. Transport RNAs are very, very particular shaped. So I'm going to bring this back here. So this one here is our transport RNA, and it's drawn really big, so it's definitely not to scale. But key features of it are that it's clover shaped. It's clover shaped, and it has 3 unpaired bases at the middle loop. 3 unpaired bases. Now these unpaired bases, I'll write that in a 3 3 unpaired bases. And this is called an anticodon. An anticodon. And it's called an anticodon for a very good reason. The anticodon matches a very specific codons. And so we know what codons are. Codons are the mRNA script for a specific amino acid. So the anticodon is the opposite code that matches the codon. And so therefore, there's communication that occurs to get the correct amino acid to be brought to the ribosome in order to be put together. So we'll go one more time. The ribosome is reading the mRNA strand 3 nucleotides at a time. Those 3 nucleotides are a codon. So one codon at a time. As it reads that codon, it summons for the anticodon to be brought to the ribosome. So the anticodon exists on the tRNA, which is attached to the correct amino acid. So the ribosome reads the codon and signals for the anticodon to connect with it and delivers the amino acid to be put together to start forming the protein. Once those amino acids start to build up at the ribosome, they form a polypeptide chain. So we end up with all of these amino acids that are all being clumped together in a nice big chain. It's not yet a protein, it becomes a protein once it begins to, so once the ribosome has read the whole mRNA strand, it will release it, and that polypeptide chain will begin to fold and twist and swivel on itself to create a very specific shape, because shape is very important for proteins. It will combine with some other chemicals in order to form a very, very specific protein. So if we come back to our diagram just that one last time, one last recap for it all. So we know that protein synthesis occurs inside the nucleus, or begins inside the nucleus with the DNA. When the DNA unwinds and unzips, and the RNA polymerase reads the DNA and pairs up complementary bases, with the exception of thymine, no thymine in mRNA. Instead, uracil is connected to adenine. So once these bases have been put together and we now have a strand of mRNA, the mRNA detaches, the DNA goes back to normal, the mRNA modifies itself, in other words, it kicks out the intron, which is the non-coded mRNA, joins together the exons, which are the coded mRNA, cuts off the extra nitrogen bases on the end, caps them, and puts tails on them. Once it caps it and puts tails on it, it then leaves the nucleus and goes to the rough endoplasmic reticulum, where it is read by the ribosomes. As it reads the ribosomes one codon at a time, or 3 nitrogen bases at a time, it will read that codon, and that will signal, the ribosome will signal for the anticodon to come to the ribosome with the correct amino acid. Those amino acids are then joined together into a polypeptide, which is lots, poly meaning many amino acids together. Once the chain has been made, it will release it, where it will fold and reshape into a protein. Hope that made sense. Good luck.